not make arginine. And they isolated a whole bunch of mutants that could not make arginine, called arginine 1, arginine 2, arginine 3. And they knew the approximate pathway for the production of arginine based on experiments that had been done by biochemists. And so they had intermediates in the pathway, and they decided to feed these neurospora these intermediates. They would include the intermediates into the agar on which the cells were growing, and the nutrients would be absorbed by the cells, and then they would ask the question, under these conditions, could these mutants grow? And so they had 3 mutants, that's called the mutant 1, 2, and 3, and they started feeding them ornithine, citrulline, and of course arginine. These are the intermediates in the pathway. And when they did this experiment, they got results that are shown here. Mutant 1, called ARG1, if they fed them ornithine, the mutant was not unable to grow. However, if they fed it citrulline, the mutant was able to grow. And if they fed it arginine, the mutant was able to grow. So they basically concluded from this that mutant 1 basically functioned downstream of ornithine and upstream of citrulline, right? Because when you feed it citrulline, it can carry on down this pathway. But when you feed it ornithine, it could not, so the mutant was blocked. There was a roadblock in this pathway right over here in this mutant. It was not able to go from ornithine to citrulline. Mutant 2, on the other hand, was able to grow when you fed it ornithine, when you fed it citrulline or arginine. And because all 3 of these were able to rescue this mutant, because that's what this is, it's a rescue of the mutant phenotype, mutant 1 must be upstream of ornithine. Because once you bypass the block by feeding it ornithine, the pathway is intact and it moves down the pathway. And then, of course, mutant 3, you could only rescue this mutant for growth in the presence of arginine. Adding back citrulline or ornithine, you were not able to rescue the pathway. So this experiment is known as chemical complementation. It's distinct from genetic complementation that we're going to talk about later on in this class. Okay? So keep that in mind. This is only a mechanism of studying mutants in microorganisms. You can't use this mechanism of feeding intermediates to larger eukaryotes, basically like Drosophila or mice or humans. You can only use it for microorganisms because they absorb all sorts of nutrients from the agar. So you can do these sorts of experiments in these organisms. Okay. So based on the results, they were basically able to say that mutant in arginine 2, the only thing that this mutant would allow to be made is the precursor. You had to add ornithine or citrulline to the medium. Downstream enzymes were still intact, and the pathway would basically continue, and you would get arginine synthesis. Mutant in arginine 1, you could supplement the media with citrulline, but adding precursor or ornithine was not sufficient because the mutation was blocking the pathway. So, what this result also shows you is that there were 3 different genes that had the same phenotype. If you look at the phenotype as synthesis of arginine, if you focus entirely on the production of arginine, all 3 mutants, arginine 1, arginine 2, arginine 3, were unable to make arginine. So 3 different mutants in 3 different genes had the exact same phenotype. And that's important to keep in mind. So when you have genes in a single pathway and you're looking at the last step in the pathway, all of the mutants in that pathway will appear to have the same phenotype. Now, of course, if you focus on production of ornithine or production of citrulline, the different mutants will have different phenotypes. But if you focus on the last step, then it will appear that there are 3 different genes or 3 different enzymes that are necessary for a single phenotype, the production of arginine. All right? So that's important to keep in mind as we go forward now. We're going to talk about genes that take part in the production in a single genetic pathway, or even a single biochemical. Okay, so this observation that there are 3 different genes, or 2 different genes, or 5 different genes affecting the same phenotype, if you were to do crosses with these mutants, classical crosses like Mendelian crosses, the phenotype ratios will be weird and different. And we'll come to that very soon when we talk about epistasis. Remember, this is the part of chapter 3-2 that we skipped before the midterm because I didn't want you to get confused with all the other variations and ratios that I was talking about. We're now going to go back to chapter 3.2 just for this one little bit of the class. But before that, I want you to keep one other thing in mind. And this is an important point that some of you forget. The mutants we are talking about and studying using the method of Beadle and Tatum by feeding chemicals, you can only do this analysis for mutants that are recessive. All right? Complementation can only be done with recessive mutants. They cannot be done with dominant mutants. So if a mutation is a dominant mutant, you will not be able to perform this kind of an analysis. Okay, so here is another pathway. All right, so you basically have mutants in this pathway. Mutant 1, mutant 2, mutant 3. And you have compounds that you know might rescue the pathway. But you don't know the order of the pathway, and you don't know at what step the mutants are functioning. Based on the rules that we just talked about from the Beadle and Tatum experiments, you can reorder this entire table so that you can figure out the pathway and figure out which step the mutants are playing a role in. So you can do 2 things in one shot from looking at this table in this chemical complementation analysis. So here are 4 different compounds, compound E, B, N, and A. And we have 3 different mutants, mutant 1, 2, and 3. Okay, mutant 1, if you feed it compound E, you're not able to rescue it. If you feed it compound B, you're not able to rescue it. But when you feed it compounds N and A, you are able to rescue the mutant. So compounds N and A must be downstream of the mutation, correct? So that's why you can rescue them. Mutant 2, you can only rescue it by feeding it compound N, but not by feeding it E, B, or A. So N is very likely going to be one of the last steps in the pathway. And then mutant 3 is a mutant where you can rescue it by feeding it E, N, and A, but not B. So mutant 3 must work between B and one of these other mutants. So when you get a table like this, there is a very simple way of transforming this table that allows you to very quickly and easily figure out the pathway and the step that the mutant's playing a role in. You basically have to take this table and transform the rows and the columns, okay, in a series of steps. And I'm going to show you these in a minute. So here is the original table. And you want to basically figure out the order of the compounds in the pathway and where in each of those steps the mutations are. So the first thing to do is you rearrange the mutants. Okay? Mutant 1, mutant 2, and mutant 3. So these rows you rearrange in this table. So you transform it in a way such that the mutant that has 3 positives is the one on the top row, followed by the mutant that has 2 positives, followed by the mutant that has 1 positive. So in this analysis, which mutant has 3 positives? Mutant 3. So you basically put mutant 3 in the top row, followed by mutant 1 in the second row, followed by mutant 2 in the third row. So you reorganize this table in this fashion. You keep the compounds the same, E, B, and A. You just retransform this table to go mutant 3, which is plus, minus, plus, plus. You keep it plus, minus, plus, plus. Mutant 1, minus, minus, plus, plus. and mutant 2 that goes minus, minus, plus, minus. That's the first step in the transformation of this table. You're not changing any of the values in this table. You're just transforming it, basically, reordering the rows in a sense, in a particular pattern that goes 3 pluses, 2 pluses, 1 plus. If you had 5 mutants with 5 pluses, you would start with 5 pluses, 4 pluses, 3 pluses, 2 pluses, 1 plus. Whatever the numbers, you just order them in a decreasing order of positive values. All right? That's the first step you do. Once you've organized the table by reorganizing the rows, then you reorganize the columns in this table. And the way you reorganize the columns is on the extreme right, you have all the strains with pluses, followed by 2 pluses, followed by 1 plus. So you're now reorganizing the columns, i.e. you're reorganizing the compounds. And so when you do this, mutant 3 still stays on top now, mutant 1 is second, mutant 2 is third. You have 3 n's right down there, because n is the one that has 3 pluses, so you move n over here, followed by the column with 2 pluses, which is A, followed by the one with 1 plus, which is an E, followed by the one with no pluses, which is a B. So the pathway is a compound B gets converted to compound E, gets converted to compound A, gets converted to the product N. N is the last step in the pathway that you're studying. Mutant 3 is required for the conversion of B to E. Mutant 1 is required for the conversion of E to A. And mutant 2 is required for the conversion of A to N. So just by rearranging the rows and the columns in these chemical complementation tables, you can figure out the entire pathway and where in the pathway each mutant functions. Simple transformation, right? Now this experiment can only be done for chemical complementation. You don't do this analysis for genetic complementation. That's something we'll come to later on. So the tables for genetic complementation look quite similar to the tables for chemical complementation. So you have to pay attention to what I have been asking you. If I'm talking about feeding compounds, that would mean it's a chemical complementation experiment. If I talk about checking one mutant against another, it's basically a genetic complementation. You don't do this transformation for genetic complementations, only for chemical complementations. Okay, now here is something else that you have to keep in mind. So here is the pathway BEAN, mutant 3, mutant 1, mutant 2. However, there's something important to keep in mind. The steps in the pathway identified by this procedure are entirely dependent on the intermediates that are available to you as an experimenter and the mutants that you have isolated. So the pathway may actually be much longer. You may not have all the intermediates in your lab, so you can only feed them the intermediates you have in the lab. So the pathway may have additional steps, but you won't detect them. This is basically a simple pathway that you can detect. In addition, you may not have mutants for every step in the pathway. You may only have mutants for 2 out of the 4 steps in the pathway. That's also possible. So it doesn't identify every step in the pathway, and it does not identify every enzyme that is functioning in the pathway. So the pathway may very well be BESAN, but you may have only identified BEAN. That's fine. At least you know some of the steps in the pathway. You can then do other experiments to figure out the rest of the pathway. But this is a start, in a sense. Okay. Also, you may get mutants, 2 mutants that affect the same step. That's also possible, because when you're mutagenizing cells, there's nothing to say you can't mutagenize the same enzyme in 2 different cells at the same, right? It can happen. It happens quite a lot. So you may identify mutant 5 and mutant 3 being required for converting B to E. The table for something like that would look like this. Okay? So you have the previous table, mutant 1, 2, and 3. You may have additional mutants, mutant 4 and mutant 5. You notice mutant 4 looks exactly the same as mutant 1. right? So mutant 1, mutant 3 and mutant 1 and mutant 4 basically function in the same step. Similarly, mutant 5, the feeding of the compound experiment, looks exactly the same as mutant 3. And so mutant 3 and mutant 5 are probably two independent mutations isolated in the same enzyme. Right? You can easily get that. Multiple alleles of a gene. You could get that. So it's entirely possible. And that also happens very often. So you may have something that looks like this, or you may have something that looks like that. Keep that in mind. Questions about chemical complementation? Okay, so this is the exact rationale, mutagenizing, analyzing mutants, that's being used recently in the lab to study all sorts of interesting biological phenomena, from the study of fly development, to the study of behavior, to the study of biological clocks, circadian clocks, to the study of aging, as well as cell cycle, etc., etc. The review, basically what you have to keep in mind is that any biochemical or biological process occurs in a series of steps. Each step is regulated by a specific enzyme, and each enzyme is made by a single gene. And so you can use this principle. You can't feed everything, but you can use the principle to figure out pathways by doing these kinds of experiments. Any questions or anything else? Okay, go ahead.
Yeah, about the rearranging the mutants and compounds, does it have to be in that order?
No. No, you could do the columns first and then the rows, or the rows first and then the columns. You'll come to the same answer. Okay, so now we're going into epistasis. So chapter 3-2, we're going back to chapter 3-2. Okay, so this is the point I made earlier. Beadle and Tatum's results showed that there were 3 different enzymes necessary for a single phenotype if you focus on the production of arginine. Right? So if you were focused simply over here, arginine mutant 3 would be unable to make arginine, arginine mutant 1 would be unable to make arginine, and arginine mutant 2 would be unable to make arginine. So what happens now if, say, you were to take an organism that had mutations in a same phenotype and you did some kind of crosses? So you're doing a Mendelian cross with mutations in, say, 2 genes or 3 genes. But the important thing is the mutations in those genes have the same final phenotype. They may have intermediates that may have different phenotypes, but they're a single phenotype at the end. So how do you basically analyze this and figure this out? So let's look at some. Corn. So let's look at what you've observed in the grocery store, basically, right? Corn. You can get blue corn, you can get white corn, you can get yellow corn, right? Basically, it's a pigment that is made in the kernel of the corn. Now, this is a very simplified part, but there are many steps involved. I've just put out the key intermediates. Okay? White, flavone, and chalcone, which is yellow. And then there's a precursor. We're going to ignore the precursor for now. We're going to keep it simple. We're going to focus on this part of the pathway. Right? So, if you buy yellow corn, where in the pathway would the mutation be? Would it be here? Would it be here? Or would it be here? And the answer is, it's going to be right there. Why? Because this conversion of yellow to white is being blocked. You're accumulating yellow in the kernel of the corn. Right? If you had white corn, then that would mean that the mutation was basically taking place right there, ignoring the precursor. Right? Keep it simple. And if you had blue corn, of course, meant there were no mutations in that plant. The plant was wild. So now let's take a corn, true breeding. Remember, we're starting with cross. So we're going to start with some true breeding corn of different phenotypes, and we're going to cross them. And the mutants that we're going to analyze are mutants that play a role in this pathway. Starting with yellow, going to blue. So we're going to look at two different mutants for simplicity. A mutant, an enzyme that converts yellow to white, and then an enzyme that converts white to blue. Right? Fair enough? So, always try and start a cross with a true breeding strain. In this case, we're going to start a cross that's going to be okay. We're going to start a cross with a true breeding strain that's white and a true breeding strain that's yellow. And then we're going to ask, what kind of progeny will we get when we do this cross? I've already drawn out the pathway for you. It's a simple pathway. You're going from yellow to white to blue. Okay, so what's the genotype of this white corn? What do you think is the genotype of the white corn? It's a true breeding strain. Let's call the enzymes A and B and the genes also A and B. So if you are white, right here, what would your genotype be for the two genes, A and B? Big A. And what kind of gene would B be? It's a mutant. We're talking about two genes here. Oh, wrong. Let me give you a minute. A white corn, true breeding white corn, right? So you're accumulating the white pigment, which means that the enzyme B is mutated, right? Little b, little b. But enzyme A is normal, big A, big A, correct? And you're crossing it to a yellow corn. The yellow corn will be mutated for this enzyme because you can't convert yellow to white. You're accumulating yellow. So you're going to be little a, and you're going to be big B, big B. Okay? Clear? Question?
How do you know that it's big B, and
I'm true breeding, so I have a mutation in a single gene. That's how you start it. Now you can have a cross. I would have to tell you this, but I can start a cross that is big A, big B, cross to little a, little b, little b. What would be the phenotype of a corn plant that's little a, little b, little b? It will be yellow, exactly. And that's where the ratios start to get muddled. And that's why I moved this section to after the midterm, because the ratios are now going to be really interesting. So I didn't want you guys to be confused with this ratios we were talking about before the midterm. So this is where we're going. All right. So we're going to start this experiment doing this cross. A true breeding white with a true breeding yellow. So here we go. In the F1, you're going to generate a plant that's going to be blue. Right? This is not incomplete dominance. This is not codominance. This is a completely new phenotype in the F1. Why is it blue? Well, your heterozygous big A, little a, so you're producing this enzyme. You can convert yellow to white. Your heterozygous big B, little b. So you're producing this enzyme, so you can convert white to blue. And so all of a sudden, in the F1, you get a completely new phenotype, which is blue corn. Blue is definitely not incomplete dominance, because it's not something between yellow and white. You can't say blue is a color between yellow and white, right? And it's not codominant, because codominance would mean that you would have yellow and white at the same time in the same cells. And that's not what blue is either. So it's not incomplete dominance, not codominant. It's a new Oxford phenotype in the F1. Okay? So very distinct from what Mendel had seen. The reason is the two enzymes and the two genes function in this pathway. And so when you are heterozygous, you're basically able to produce the final product. Okay, so now let's do Mendel's self-cross. You take this blue F1, cross it to itself. What would you get? What's the classic Mendel experiment of a dihybrid cross, self-cross? When you take a heterozygous individual to a heterozygous individual, what's the ratios you would expect? 9, 3, 3, 1, right? So let's see what you get now in this cross. Okay, so this blue individual will produce 4 kinds of gametes, correct? Let's assume at this point that these two genes are assorting independently. They're not linked. We're going to keep it simple. We're going to say that the two genes are assorting independently. Right. So you're going to get 4 different kinds of gametes produced by this heterozygous blue corn. Big A, big B, little a, little b, big A, little b, and little a, big B. So let's write out those 4 gametes for one corn, and then we're doing a self-cross. So we have them here, right? Okay, now these gametes will fertilize one another. What will the color of this corn be? Big A, big A, big B, big B. Blue. Correct? Because you're making enzyme A and you're making enzyme B, they're going to go all the way to blue. The next one will also be B because will be blue because you're big A and big B. The third one will be blue because you're big A and big B. And the fourth one will be blue because you're big A and big B. Again, big A and big B, blue. But now what happens here? Your big A, little b, little b. What phenotype would big A, little b be? So your big A, but your little b is going to be white. Right? So you're taking a blue corn, crossing it to a blue corn, and now you're getting blue and white. White's a completely new color in a sense. Then you get blue again, and then you get another white, big A, little b. And then big A, big B will be blue, big A, big B will be blue, but then here, I don't know if you'll be able to see this, but let's try. This is basically little a, big B, big B. So you are little a, so you're blocked over here with your big B, and you're going to be yellow. And then this also is going to be yellow. And then your blue here, white, yellow, yellow. So the ratio out of this cross, of a blue self-cross to a blue self-cross in the F1 will be 9 blues, 1, 2, 3, 4, 5, 6, 7, 8, 9 blues, 3 whites, 1, 2, and 3 whites, and 4 yellows, 1, 2, 3, and 4. Now, it's not 9, 3, 3, 1. The reason is this 1, which is little a, little b, will be yellow because it's basically being blocked here. So even though this enzyme is not functional, the phenotype you observe is going to be yellow. Right? Because this enzyme works upstream of this enzyme. It works before the second enzyme has a chance to work. And so this downstream effect is being blocked, is being masked by the recessive mutations upstream. The recessive mutations little a is masking the effect of a little b. This is known as recessive epistasis. Okay? Mutation in this gene that's working ahead of the mutation in this gene is masking the phenotype of this mutation. This is a recessive epistasis. So when you do a self-cross of a heterozygous to a heterozygous, and you get a ratio that goes 9:3:4, you basically can conclude two genes that are playing a role in the production of the blue pigment And most importantly, you are actually able to tell the pathway. You are able to indicate that yellow is the precursor, white is the intermediate, and blue is the product. The one that gives you 4 is the precursor, the one with 3 is the intermediate, and then the one with 9 is the final product. Questions about this?